well as giving out the International Gairdner Awards, the Gairdner Foundation considers it's part of its role is to really not just celebrate excellence in science by prizes, but celebrate excellence in science wherever it exists and translate that into delivering scientific information to the public, to high school, to policymakers, to universities, and really engage with the best scientists around the world to encourage and point out the importance of science in society today. As part of what we do, we have started running these global perspectives panels over the last year in a virtual world that we now all live in. And we've covered a number of important topics relevant to Canadians today. One of the topics that you cannot avoid, of course, is the COVID situation. And a number of our panels have addressed that. And today's is going to be quite a special one because what we're going to do is to look at some of the companies in Canada that are working at the front line in developing therapeutics and vaccines for COVID and where they stand today and their success stories. So this is a story of Canadian success on the international stage. The title of our Perspectives panel is COVID Treatment and Vaccine Development. And we're very lucky to have with us 3 people from 3 different companies to talk about their work in this area. We have Véronique Lecault, who is co-founder and chief operating officer at AbCellera in Vancouver, and I'll tell you more about her in a moment. We also have Tom Madden, who is the CEO, I think, sorry, it's not on my list, you are the CEO, yes, of Acuitas Therapeutics in Vancouver, and he'll tell you about his technology as well. And then finally, Brian Ward is joining us from Montreal, where he, among other things, is the medical officer for Medicago Incorporated in Quebec City. So we'll hear more about their research and we'll have an opportunity for question and answer. At this point then, I'm going to begin by turning to Véronique to give our first presentation. And let me tell you a bit more about Véronique. So Véronique is co-founder, chief operating officer, and member of the board of directors at AbCellera. She received her PhD in chemical and biological engineering from the University of British Columbia. So when she was at UBC, she co-invented the high throughput screening platform that is now part of AbCellera's core technology. It combines microfluidics, hyperscale data science, machine learning, bioinformatics, and genomics. AbCellera searches, decodes, and analyzes natural immune systems to find antibodies that can be developed into drugs to prevent and treat disease. And of course, in this particular instance, we're going to hear about their approaches to finding antibodies to counter COVID. Véronique was recently recognized as a STEM star influencer by BC Business and is one of Canada's top 40 under 40. The presentation title is Combating COVID-19, Identifying a Potential Drug Candidate for Human Testing in 90 Days. Véronique.
Thank you very much, Dr. Rossant. It's a pleasure to be here today and tell you more about the work that we are doing at AbCellera. In 2018, Bill Gates said, the world needs to prepare for pandemics in the same serious way it prepares for war. And now here we are 3 years later with over 100 million cases of COVID-19 around the world and more than 2 million deaths. And at AbCellera, we had been preparing for over 2 years before the current pandemic unfolded. We were selected to be part of the Pandemic Prevention Platform program called P3 from DARPA, that is part of the US Department of Defense. And for those who are not familiar with DARPA, they are known to fund bold ideas that sometimes approach science fiction. So they have been credited for being behind the invention of the internet and GPS technologies. And the goal of the P3 program is to develop an antibody countermeasure that can be field ready in 60 days to stop a pandemic from the time a new virus is identified. And to adapt our platform for pandemic preparedness, we had gone through a few tests and did some simulations to learned how we can quickly discover antibodies from samples from patients who have been infected with a virus. And then on January 28, 2020, we received a phone call from the NIH and learned that the first case of COVID-19 was confirmed in the United States. And this is where the team quickly mobilized. And a few weeks later, we received access to a sample from one of the first patients in the United States who had recovered from COVID-19. So this patient who had recovered from the infection had developed molecules that we call antibodies. And we all make naturally billions of different antibodies. This is nature's natural response to fight diseases and infections. And each of those antibodies are made by a subset of cells in our immune system called B cells. And almost every cell makes a different antibody. So there are in our immune system some antibodies that have the right properties to be turned into drugs, but finding them can be sometimes challenging. So this patient who had recovered had a neutralizing antibody. And when we received the sample, 4 days later, we started screening those blood cells. In 3 days, we looked at 5.8 million different single cells using our Microfluidics technology. And this is a technology that was originally developed here in Vancouver at the University of British Columbia. We use chips that are the size of a credit card that have many, many chambers, very small in volume, so 1 nanoliter each. So it means that we can put these single cells into those chambers, and because they're so tiny, we can quickly concentrate the antibody that are being produced by those single cells and then identify which cells make antibodies that are specific to the antigen. And in this case, that was SARS-CoV-2. And those chambers that have the cells we're interested in, they essentially glow under the microscope, and we can then recover them and look for the genetic sequence of those antibodies. So we recovered 2,200 single cells in 3 days, did next-gen sequencing to find the sequence. So that's essentially the recipe that allows us to produce those antibodies, either at small scale to do additional testing or at larger scale for manufacturing into a drug. So we identified through this process 500 unique different antibodies. That was 9 days from receiving the sample, and then expressed some of those to do additional testing. We had over 250,000 data points to analyze, and we looked at which antibodies had the best ability to neutralize the virus, but also which ones had the best properties to be turned into successful drugs. And then we selected this down to 24 lead antibodies using Cilium, which is a custom bioinformatics software that we've developed in-house to be able to analyze those massive data sets when we look into this natural database that are immune repertoires. So from day 0 to day 90, we had identified the lead antibody candidate, and it had entered human clinical trial with our partner, Eli Lilly. And to put things in perspective, in normal times, it would take typically 10 years to bring a new biologics drug to the market, and at least a few years to bring it to human clinical trials. So currently, Vamlanova has received emergency use authorization in the United States, as well as interim authorization by Health Canada. It has been shown to reduce the risk of hospitalizations by 72% in patients who have mild to moderate disease and who are at high risk of complications. We've also recently received additional data from those clinical trials done by our partner, Eli Lilly, and these data have been announced publicly, confirming in phase 3 clinical trials that Vamlanova can reduce hospitalizations by 70 to 80% in patients who have been recently diagnosed with COVID-19. It can also prevent infections in nursing homes and reduce the risk of contracting the disease by 80% in residents who may be exposed to the virus. And in that same trial, Vamlanova has also been shown to prevent death. So it's really a shield that has been proven to be effective against hospitalization, infection, and death for those who are the most at risk of complications from COVID-19. It's currently approved in 8 countries, and it has been administered in over 125,000 patients so far in the United States. And to be able to accomplish that, it takes a big team. Of course, the AbCellerites who have put their heart and soul into this program. We have engineers, data scientists, bioinformaticians, scientists, project managers, and the business professionals who are supporting this effort. And we've been fortunate to partner with Eli Lilly and Company as well as a Vaccine Research Center, and DARPA, of course, who have funded the original work on pandemic preparedness that allowed us to be really ready to respond at the time where the world needed it the most. And we've also been working with the McClellan Laboratory at the University of Texas. So at AbCellera, we've developed this platform that allow us to source, search, find, analyze, and engineer antibodies. And we're really currently at the early stages of discovery, and we work with partners on all sorts of indications and essentially deploy our technology towards different targets, but it's still very early stages. And in this case, we partner with Eli Lilly, who have the ability to run the clinical trials and manufacture those antibodies, as well as do the distribution. Currently in Canada, we do not have the capabilities to produce clinical grade monoclonal antibodies at large scale. And we've been fortunate to receive support from the government of Canada to build a GMP manufacturing facility that will allow us to be better prepared in future pandemics and have the ability to go from discovery to making enough antibodies for clinical trials. And finally, to be able to be prepared when there is the next pandemic. And unfortunately, it's possible that we will see more of those in our lifetime. The surest path to a better future is to continue to invest in technologies, to continue to invest in our talent and in teams, and to build the research infrastructure in Canada that allows us to do this important work.
It's a very interesting story, and again, the rapidity with which you've been able to go from the serum to the monoclonal is quite remarkable, and obviously has a lot of potential, not just for COVID, but for many other disease indications in the future. So I can see why there's a lot of excitement around the AbCellera platform. Thank you. So now I'm going to turn to our next speaker, which is Tom Madden. And Tom from Acuitas Pharmaceuticals got his BSc and PhD in biochemistry from the University of London, and he's held several senior academic and industry positions over his career. I won't go through them all. He founded Acuitas Therapeutics in 2009 and has really guided the company to a leadership position in the application now of lipid nanoparticle technology for delivery of nucleic acid therapeutics. So now we're going into the nucleic acid area. And recently, Acuitas has focused on the development of LNPs for messenger RNA therapeutics. And this, of course, includes the carriers used in several COVID vaccines, including particularly the Pfizer-BioNTech vaccine, which we all know, of course, was authorized in a number of countries, including Canada, and is in use in Canada in our vaccine interventions. So again, going from discovery in the lab, as was the case with AbCellera, discovery in the lab through to therapeutics that are being used practically in delivering therapies today. So Tom's title is Canadian Technology Enabling COVID-19 Vaccines. Tom.
Thank you, Janice, and thank you for the invitation to present today. So just to start, I wanted to mention that I'm really taking very much a 30,000-foot level approach to the topic, and my talk is really intended for a very broad audience. So my apologies in advance if I don't go into the technical details that you might like to see. So I wanted to start by acknowledging one key aspect of the timeline around the COVID-19 pandemic. And the first reports of this disease, not then called COVID-19, came out in December of 2019. And interestingly, the virus had been isolated and its genetic sequence determined by January 10th. And so this illustrates the power of genetic analysis to very quickly allow us to understand the makeup of a viral threat or an emerging viral threat. And with the genetic analysis for COVID-19, we were able to determine what proteins form the virus, and in particular to identify that, as with other similar coronaviruses, it has a spike protein associated with the surface of the virus that is required for the virus's infection of our cells. And that's sort of illustrated on the next slide here, where the spike protein binds to a particular protein or receptor on the surface of human cells, the ACE2 receptor. And it's this interaction which is required to allow the virus to infect our cells. And so clearly therefore, the spike protein is an excellent target for a protective vaccine, as discussed in the presentation by Véronique. Now, the technology that we've been working with our partners is using a new approach to the development of vaccines. It's using, it's called messenger RNA technology. And I wanted to first of all start off by just explaining what messenger RNA is to those of you whose expertise may be outside of biology. So within our cells, proteins are considered sort of the workhorses of our cells. They do most of the work in the cells. They catalyze most of the reactions, and proteins form most of the structures within our cells. And these proteins, there's tens of thousands of them, and the information on how to make them is contained within our genes, within the DNA. Now, as you probably know, our genes, the DNA is inside the nucleus within the center of our cells. And so when the cell wants to make a particular protein, the information for that protein has to be transferred from the nucleus into the cytoplasm of the cell so the protein can be made. And the message that tells the cell how to make this new protein is called messenger RNA, and it's the instructions to make a specific protein. Now, what was sort of recognized several years ago is that if we can deliver a messenger RNA that provides the information to make a viral protein, then our cells will generate that protein, and this should then trigger an immune response against that protein that would protect us should we be exposed to the virus in the future. And so this is a very exciting, very elegant, precise technology to potentially generate a vaccine. But there are a couple of important problems. And one is that mRNA, when it's administered to our body and is outside our cells, is very, very quickly broken down. It's very, very labile. And compounding this problem, it can't get into our cells by itself. It's a huge molecule, and so there's no way it can enter cells by itself. And so we needed to develop a delivery technology that would protect the messenger RNA after it's administered, and more importantly, carry it into our cells and make it available within the cells so that it can be translated into the protein. And I'm going to try and quickly show you a video that illustrates how our technology works. And so, first of all, I wanted to point out that the messenger RNA here is shown in yellow. And as you can see, it's encapsulated within our delivery technology, microscopic lipid spheres. And these protect the mRNA after it's administered and prevent it from being broken down. And then as I run the video, you'll see how they carry the mRNA into our cells, release it to the cell interior so that it can be translated to give rise to the protein shown in red. And I'm going to have to, So the idea, as I say, of using mRNA as a vaccine has been around for several years now. And we've been supporting partners on development of these vaccines prior to COVID-19 coming along. And what's sort of interesting here is that the sort of coincidence in timing for this earlier work we were supporting is really very fortuitous. So we've been working with one of our partners, a company called CureVac in Germany, on a rabies vaccine using our delivery technology with their messenger RNA, and had been conducting a phase 1 trial in 2019. And right at the start of the emergence of COVID-19 in January 2020, the results from that phase 1 trial became available. And what we saw was that the messenger RNA vaccine against rabies could generate a very strong immune response at remarkably low doses. I almost think of these as homeopathic doses. And so this clinical data was very exciting. And as I say, it was generated just as the threat from COVID-19 was being recognized. And so we then quickly engaged with several of our partners, including biotech, who were partnered with Pfizer and CureVac, with whom we were working on the rabies vaccine, in order to develop vaccines against COVID-19 using the same technology. And this work has been, fortunately, very successful. So our partner BioNTech and Pfizer received authorization for their vaccine. It's called Comirnaty. It was authorized for emergency use in December of last year. And CureVac have initiated a late stage clinical trial. It was initiated again in December last year, and they're expecting to submit for regulatory authorization for emergency use in the first half of this year. I wanted to sort of now sort of quickly turn to one of the pressing issues at the moment, and that's the emergence of COVID-19 variants, and just explain a little bit about what these variants are and how messenger RNA vaccines could potentially address them. So I mentioned that proteins are the sort of the workhorse. We're developing a vaccine that's targeted against the spike protein on COVID-19. And proteins consist of building blocks of amino acids, of which there are 20 different amino acids. So the COVID spike protein has a total of 1,273 amino acids. And as I say, these create the final protein. And variants arise, and the variants we're talking about specifically at the moment are variants where certain amino acids in the original sequence have been changed to a different amino acid. And that's sort of illustrated by the colored spheres in the diagram on the right-hand side here. So these variants can make the virus more easily transmitted, and they could also cause more serious disease. And so they're a key concern at the moment with respect to the potential use of vaccines. And so first, I wanted to just note that the current mRNA vaccines express the original sequence of the spike protein. And it does appear from preclinical research that's been conducted recently that the BioNTech Pfizer vaccine continues to be effective against the UK and South African variants of COVID-19. So that is good news. But I think we need to be aware of the fact that variants could potentially arise where the current vaccines are not protective. And this obviously would be a very serious situation. What is really attractive about the use of messenger RNA for a vaccine is that we can very easily revise the message sequence to exactly express any new variant. So those changes in the amino acids I talked about can be encoded in a new message, and so we can develop versions of the vaccines that are already authorized, which exactly express the new variant and protect against it. And in addition, if we wanted to, we could include multiple mRNA sequences within a single vaccine that express multiple versions of the COVID spike protein. And again, that should provide, broad protection against a range of different variants should they emerge in the future and not be addressed by the current vaccines. And I will stop there and thank you for your attention.
Thank you very much, Tom. That was a very clear presentation. I'm looking at the Q&A, and it's interesting to see that some of the questions that have been coming up have been sort of answered as we go through the presentations, but we'll come back to the questions at the end. So now I'm going to turn to Brian Ward, our last speaker today, who's going to present an update on Medicago's plant-derived vaccine. This is a totally novel technology, but also one that's moving into clinical trials and shows some exciting opportunities, I think, for the future. So Brian Ward was trained at McGill and Oxford. He was a Rhodes Scholar at Oxford and at the University of London, Johns Hopkins. He returned to McGill in 1991, and he's currently a full professor in infectious diseases and tropical medicine. And he's carried out many different roles nationally and internationally in matters related to global health and vaccines. He was elected to the Canadian Academy of Health Sciences in 2012, and he chairs the Scientific Advisory Board for the Institute of Infection and Immunity of the CIHR. And since 2017, he's also served as medical officer for Medicago Incorporated. And he tells us that he's on a sort of part-time leave from his position at McGill and has been doing that for 3 years and probably further as the company goes forward with its products. So Brian, please update us.
Well, so many thanks, Janet, and to the Gairdner Foundation for the invitation to present at the symposium today. This picture that you see is the sort of the unique environment in which we produce our vaccines. Instead of using huge stainless steel fermenters, we actually use the plants as our bioreactor. Our vaccine is actually a fairly traditional vaccine because it's protein and adjuvant, but the platform itself is really quite new. So I'll just very quickly walk you through Medicago, then talk about the vaccine, and then tell you where we are. And rather than Tom's 30,000 foot, we are very much in the weeds right now as we move rapidly towards the introduction of our phase 3 clinical trial. So Medicago, very quickly at a glance, we've been working, starting in a double-wide garage 20 years ago. And we have production facilities in Canada and the United States now, and we're building a global facility. That will be my last slide in Quebec City. We currently have 450 employees in Canada and the United States, 56% of whom are women with a very diverse workforce. There are 5 vaccines in the clinical pipeline, and so far we've actually conducted studies mostly with pandemic and seasonal influenza vaccines, including more than 27,000 subjects. And I have to say, it's fairly daunting to be a company with 450 employees, often on the same stage as the Pfizers and the J&Js of the world. The plant manufacturing process is straightforward, and it's a recombinant platform where we can start with the gene of interest, in this case, the spike protein of the SARS coronavirus 2. And we then modify that spike protein so that it's in a pre-fusion form, which is the most immunogenic. We then sneak the gene into the plant leaves using a process called agroinfiltration or vacuum infiltration, where we transfer the gene using a defanged bacterium called Agrobacterium tumefaciens into the plant, which we use. It's an Australian relative of the tobacco plant called Nicotiana benthamiana. We then put the plants back in a greenhouse and let them incubate for a couple of days. They grow and they're quite happy. When we harvest the leaves and we purify the VLPs using a number of standard industrial processes, and the virus-like particles that result form spontaneously at the interface between the plasma membrane of the cells of the leaves and the plant cell wall. And so what we end up with is an envelope virus-like particle that's quite a complex structure with the recombinant protein, in this case, the spike protein, as trimers embedded into the lipid bilayer. These are very stable structures. And in fact, the cold chain requirement for the SARS vaccine is just refrigerator temperature, which makes it quite attractive. Basically, the VLPs as a vaccine platform have been in use for decades and represent some of the most effective vaccines that we have in current commercial use. So for example, hepatitis B and the human papillomavirus vaccines are both virus-like particles, not made in plants, but they're virus-like particles. So what does our candidate vaccine actually look like in real terms? And here on the left, you can see an electron micrograph of one of these virus-like particles, and it has the trimers studded around the surface, which forms this crown, which is where coronavirus actually gets their name. Below it, just slightly to the right, you can actually see images of the real virus. And you can see why the immune system might actually be a little bit confused when it sees virus-like particles, because they do indeed look just like the virus in many respects, but don't have any genetic material in them. To the right, you see a cartoon of the spike protein, where we use a full-length spike protein that has been modified to keep it in its pre-fusion form, because when the spike protein actually is cleaved to enter into the cell, it radically changes its shape. And so the pre-fusion form is the form that is most likely to be able to prevent that interaction. So I put up this complicated slide to give you some idea of how quickly we've been moving, just like Véronique and Tom. Normally, that bar across the top would extend out 5 to 10 years at least. But in fact, we have gone from the idea to preclinical development to the non-clinical animal trials through the phase 1, 2, and we are rapidly moving towards the phase 3 study in the next several days, in fact. So we have moved, you see the star up at the top. We have data from the phase 1 study that I will show you. We are almost finished with a phase 2 study, and we're analyzing the data right now. And several of these cohorts, the healthy adults, the older individuals, and the adults with comorbidities will be moved in an asynchronous way into our phase 3 study of probably close to 30,000 subjects, which will be launching really in the next 2 to 3 weeks. So this normal development program of 10 years, sometimes 15 years, has been compressed down into under a year. So highlights of the non-clinical phase 1 data so far is that we've done obviously a number of studies in mice and a large study in non-human primates, Indian macaques. And basically, we've done several GLP safety studies and immunogenicity studies in mice, no real safety issues. We've actually vaccinated non-human primates and challenged them with no evidence of vaccine-enhanced disease and with good evidence of protection. In our phase 1 clinical study, we looked at 3 different doses of our vaccine, which we're in fact calling co-VLP, 3 different dose levels, and we tested it with 2 different adjuvants, GSK's pandemic adjuvant, or AS03, and Dynavax's CpG 1018, which is a nucleic acid type adjuvant. And we tested 1 or 2 doses. The study, which I'll show you the net results of, allowed us to select the lowest dose of the co-VLP with GSK's pandemic adjuvant. And we did need 2 doses, which you'll see in the next slides. And here I'm only showing you the results from the phase 1 study of the dose and formulation that we selected. And on the left-hand side, you see the antibody responses. So an anti-spike binding ELISA. In the middle, you see a pseudovirion neutralizing assay. And on the right-hand panel, on the left side, you see a wild-type neutralizing assay. Day 21, day 42, after first and second doses in red, and in gray, you see a panel of convalescent serum, including at least 1/3 of the subjects who were hospitalized or in the ICU with very severe COVID, and those individuals have the highest antibody levels. And you can see very clearly that all 3 of these assays demonstrate a good immune response after 1 dose, so the 21-day, with a very powerful booster response that results in antibody titers that are 10 to 14 times higher than those seen in the convalescent sera. And this is very important, we think, because this is likely to be a correlate of immunity, at least in the short term. Longer term immunity is likely to require a cellular immune response. And so, of course, we measured cellular immune responses as well. And the first readouts from these assays are ELISpots. On the left-hand side, you see the interferon response. And then the right-hand side, you see a IL-4 response. Once again, you can see that at day 21, we have a good response above the background. And interestingly, on day 0 with the gamma interferon response, you actually see quite a few people who are mounting some level of response. It's low, but it's there. And that's likely to be cross-reactive T-cell responses to prior exposure to the human circulating coronaviruses. However, at day 21, you see a nice boost after the first dose and an even greater response after the second dose, both on the side of gamma interferon and on the side of IL-4. So we have a balanced Th1, Th2 response. And the Th2 response that we see is actually, we have further information, which I'm not showing you, which shows that this is actually a very powerful T helper follicular response, which is very important for the maintenance of long-term antibody production and immunity. So we are transitioning now from our phase 2 to phase 3 with this antigen dose that we've chosen, the 3.75, with AS03 as the adjuvant. We are virtually certain that we will need 2 doses and that each of our study populations moving through the phase 2 will be transitioned asynchronously as we generate data into our phase 3. So healthy adults, healthy older individuals, and then individuals with comorbidities that put them at high risk for severe COVID outcomes. And all of the ongoing COVID vaccine studies have to be enormously flexible because of what Tom was talking about with these new variants, as well as the rollout of licensed or emergency use vaccines in other countries. So we have had to significantly modify our strategy as we move forward. It's given us headaches, but it will be a very interesting thing. This is a slide that I've shamelessly pulled from Peter Dull's presentation at the January 28th COVAX meeting. And it's the first comparison across the different vaccines. And you can see here some published and unpublished data from a number of different vaccines. And I want to bring your attention. Each of these vaccines compared themselves to a panel of convalescent sera, as we did. And you can see at the top the relative fold increase above or below the convalescent sera. And below, you can see the vaccine efficacy that's been published by these different vaccines. And it's very clear that as you increase the fold higher level of neutralizing antibodies that are induced by these vaccines, you see a higher and higher level of efficacy. This is clearly true for some of the variants, that for some of the variants, the higher your neutralizing antibody titer, the more likely you are to be able to deal with some of these new variants. And you can see on this next slide, I've just abstracted our neutralizing antibody assay data from the phase 1 study, and I can tell you that our phase 2 data with much larger numbers of individuals has largely confirmed this, that in fact plants, Medicago's plant-derived virus-like particle actually elicits 10 to 14-fold higher antibody levels than convalescent sera. And well, you'll have to wait for the next couple of months to know how well this vaccine works against both the typical Wuhan-like strains as well as the new variants. So I put up the timeline again now just to show you where we are with the phase 3 study. Our phase 3 study will be like the others. It will be an event-driven study. So we will be collecting 160 cases over the next several months. We obviously hope that we will achieve a vaccine efficacy that's much higher than 50%, and we are quite confident that we will do so. It's a large study of 30,000 subjects in at least 8 or 9 countries in Canada, the United States, Latin America, and several countries in Europe and the UK. We have had to put in a blinded crossover for our study because of the rollout of the vaccines and the fact that it is no longer ethical in many cases to give a placebo, which is a requirement for some of the regulators. And so we have to put into our study, we have had to put into our study this idea that at the moment that our vaccine demonstrates its efficacy, we will cross over the placebo recipients to receive our vaccine. So at this point, just in summary, the key messages and conclusions, the non-clinical data suggested that our vaccine was highly immunogenic and safe in mice and non-human primates with strong neutralizing antibody responses, and in the non-human primates, a clear Th1 biocellular response. In phase 1, the vaccine was well tolerated. Most adverse events were mild to moderate, although they did increase with the second dose, like many of the other vaccines. A very good antibody response with the first dose, but much stronger with the second dose, and a balanced Th1, Th2 response, which we think bodes well for long-term immunity. The vaccine characteristics are quite promising because it's stable at 2 to 8 degrees, and we're using a very well-characterized adjuvant that has a large safety database. AS03 has been used in young children as young as 6 months of age, and the database for pregnant women is actually 300,000. So unlike virtually all of the other vaccines that are in development or actually have been licensed to some extent, we actually have a fairly high level of confidence that our vaccine will be safe in those unique populations. So when might our vaccine become available? Well, we're hoping that sometime in July and August. That timeline you see at the top, the star is moving as we speak, but it's probably late July or early August. And with rapid approval, efficient enrollment, and continued high transmission, we expect that we will actually be able to demonstrate efficacy in the mid to late spring, allowing us to apply for emergency use authorization in the late spring or early summer. How much vaccine can we produce? Well, we have 3 production facilities. In Quebec City, we are modifying our small pilot plant to be able to produce doses. We have our large commercial facility in North Carolina that was built with money from DARPA about a decade ago. And as you can see at the far right, our global commercial facility is under construction. Unfortunately, it will not be ready until probably late 2023, 2024. But at the current time, we have the capability of producing about 80 million doses in the next 12 months, which we can probably increase to about 190 million doses by 2022. And when the global facility is constructed, we will be among the larger companies to produce pandemic products with the ability to produce about 1 billion doses in a 12-month period. We have to thank both the Quebec and the Canadian government. You're probably aware that the Canadian government gave us $173 million just about a month ago to speed up the construction of our facility and to support some of the clinical trials. And of course, all of our other collaborators, the investigators and the participants who have participated and are participating in our clinical trials. And I'll stop there. Thank you very much.
Thank you very much. Now, if I can get all the panelists back on screen and back on video. I think those are great presentations. It's given us something to think about as we go from using antibodies as treatments to developing the tools to develop vaccines, different approaches, messenger RNA vaccines, virus-like particles, but obviously a lot of progress in all areas. Got a lot of questions. So I will try to sort of go through them as much as possible and direct them where I think they should be addressed, but I think all of you may have things to say. And perhaps the first one that's got voted to the top is something for all of you to comment on. It comes from David Rose, and it says, I've been puzzled all along there's been so much emphasis on vaccines rather than therapeutics, or at least that's my impression. Vaccines are critical, but therapeutics would have the advantage as an immediate response rather than having to treat the whole population. Thoughts? So I'll obviously, Véronique, you've been developing treatments, others been developing vaccines. It is obvious we need both, but has there been enough emphasis on developing treatments?
Yeah, this is a great question. I mean, these are really complementary approaches. Like this virus is, it's changing all the time. We have to, look at this problem from multiple angles. The vaccines have the advantage of being able to be distributed at large fraction of the population. Therapeutics, when we're looking at antibodies specifically, the manufacturing capacity is more limited in the world. So we could not produce enough for everyone to receive antibodies, for instance, as a prevention tool. But they're really complementary because if you get sick, it's too late to receive a vaccine. And also if there's an outbreak, it's also too late to receive a vaccine. It takes a few weeks to mount an immune response. So antibody therapies can act as, providing immediately that synthetic immunity for those who didn't have the opportunity to receive a vaccine yet. So I think, both approaches are going to be complementary, and as the virus evolves, we'll develop new tools as well, and it may escape, and then we can come in with different types of antibodies as well as different vaccines as well. But we will have to have several tools in our toolbox to be able to stay ahead of this virus, and I think both therapeutics and vaccines are going to play a really important role in the fight against COVID-19.
There are several questions. I'm going to stick with you for a moment, Véronique, because there are several questions related to the issue. You talked about the clinical trials, the application of the antibody in the clinic, the application in long-term care homes, which is a really exciting one. Everybody has noticed that your map is all pictures of things happening in the US and not in Canada. So the question is, why not clinical trials in Canada? And what is the future of using your therapeutics in Canada in some of these high-risk populations?
Yeah, great question. So on the note of clinical trials, in order to get the data as quickly as possible, it's also useful to run the clinical trials where the patients are. And all of this work has been done by our partner, Eli Lilly. So I cannot necessarily comment on their strategy, but the network and the patients to do those clinical trials were mostly in the United States. Luckily in Canada, we had less cases, although we do have a number of cases. So in terms of distribution, so now Vamlanova has received interim authorization by Health Canada. The government of Canada has procured 26,000 doses. We also have results from phase 3 clinical trials that confirm the efficacy that we saw in the phase 2 clinical trials in terms of reducing hospitalizations by 70% to 80% in those patients who have mild to moderate disease and who are at higher risk of complications. And then we need to find out how can we distribute this drug in the Canadian healthcare system. So there are challenges. It is an infusion, so it takes time to deliver. Eli Lilly has developed new protocols where Vamlanova can be delivered in less than 16 minutes. So there is, there is progress being made, but we have a different, we have a different system in Canada, and we certainly hope that everyone who has the ability to bring this drug to patients as soon as possible are doing everything that they can, and we hope we'll be able to also have Canadians benefit from this therapy as soon as possible.
Great, thank you very much. I'm going to turn to Tom now from Acuitas. A couple of questions here. One from Thomas Lavery, who says he understands that, not just BioNTech, but CureVac and Imperial College, you talked about some of these, are using your approach. Are the LNPs components the same in everybody's vaccine? In other words, have you got the sort of the generic version that is applicable to everybody's messenger RNA, or are there variations as you develop the products?
The issue here is we'd have to disclose confidential partner information to answer that. But shall I say it would appear sensible to use a similar composition and components in all of these.
Okay, one of the other questions related to the messenger RNA vaccines, two really. One that I actually always ask myself, because I don't really know, we put a messenger RNA in, which cells are actually making the protein, and how does the protein get put onto the cells and recognized by the immune system?
So it's a good question. I mean, and the particles are taken up by a variety of different cells in the immediate vicinity of the muscular injection, including muscle cells or immune cells. It's then processed, and parts of the protein are presented on MHC molecules, and it's likely that immune cells in the draining lymph nodes are initially those that are recognizing the foreign protein and triggering the initial.
Okay, great. So thank you for that one. I'm now going to turn to Brian, and there are several questions about your technology. One is really just the sort of cost of production. Is this a cost-effective way? Why is this a good way to make vaccine particles?
It's a great question. It's the capital expense to build the facility is actually much lower, and so the initial costs are lower. Once you actually start production at scale, there's the potential for costs to be lower as well. But it's obviously a brand new technology, and so we're only just we're the first ones to be trying this at scale. And so I guess the correct answer, or the best answer to that question, is we'll tell you in 5 years. We think it will have some advantages, and certainly in terms of the capital cost of setting up the facilities, but they still are pharmaceutical manufacturing facilities and subject to all of the same controls as the other recombinant technologies. So it certainly is not cheap to build the facilities, but over time it may well prove that the cost of goods is lower than seen in other, for example, stainless steel fermenting facilities.
Mm-hmm. I think it's a fascinating, so just to think about, it's a totally different way of thinking about production. You mentioned, Brian, that actually because your virus-like particles use the sort of well-known adjuvant, which has been tested in pregnant women and in children, that you're confident that essentially your vaccine should be easily used in a broader section of the population. Phil Marsden asks all panel members, but I'm going to direct it to you because we're running out of time. Others may comment on vaccine choices for immune deficient patients, such as kidney transplant patients. And do you see, because you're using a more standard protein and adjuvant approach, that it will be applicable in immune deficient patients?
Well, we're very hopeful that it will be applicable. The global experience with some of these other novel technologies, the RNA and DNA technologies, is expanding at an extraordinary pace. And so, the fact that AS03 as an adjuvant has been used in millions of people, these RNA and adenovirus techniques are catching up very quickly. But I'd also like to go back to Véronique's point that in fact the therapeutic and the therapies and the vaccines are very complementary, so that in many instances the therapies might keep somebody alive through their first episode, and then a vaccine might actually help to prevent them from getting sick again, because we now know that with the South African variant, for example, that many of the people who were included in Novartis's recently reported study, they had had COVID before with one of the earlier variants, and they still got quite sick with the new South African variant. So we're going to have to be looking at this sort of, for immunocompromised people, I think we're going to have to be looking at therapy followed by vaccine for many of them when they get sick.
Okay, so I think that's a good sort of wrap up, because it tells us that the kind of approaches that you're all pursuing are complementary, and that we're going to see a lot of great advances over the next while. I'd like to thank all of you. I'd like to thank the audience. There's a lot of questions still unanswered. We might collect those up and email them to you guys if you don't mind, so that if there are specific ones that you could respond to, we will channel them back. But I really want to thank you for a very enlightening and an exciting panel presentation. So thank you very much indeed.